In this lecture, we're going to talk about the microscopic anatomy of muscle cells. So first, some terminology. The cellular membrane of a muscle cell is called the sarcolemma. Remember that sarco prefix means flesh and it's specific to muscle typically. And then inside of the muscle cell are structures called myofibrils. And then instead of the endoplasmic reticulum like you would have in normal cells, it's a specialized sarcoplasmic reticulum in muscle cells. And we'll talk about its function in just a moment. So if we zoom into a muscle cell, you can see the sarcolemma, that's the cell membrane of the muscle cell. And then inside of that muscle cell, you can see there are several nuclei, because remember, skeletal muscle is multinucleate. So all of those are nuclei from one muscle cell. And then within each muscle cell, you have these structures that are called myofibrils, and they are the contractile units of a muscle cell. Within the muscle cell, the myofibrils are aligned to give distinctive bands. So there's kind of a striped pattern or a banding pattern. The I band is known as the light band, and that light band contains only thin filaments. And then the A band is the dark band, and it contains the entire length of the thick filaments that are found in muscle cells. So we'll talk about those two different types of filaments in just a minute. So those two different types of filaments are the thin filament and the thick filament. The thin filament is made of a protein called actin, and the thick filament is made of a protein called myosin. And so the myofibril is composed of both of these proteins, and they're organized into these bands. So there's in a myofibril the I band, the A band, and then there's also something called the Z disc, where the actin filaments are attached to one another. And there's also something called the H zone. So we'll go over what all of those things mean when a muscle cell contracts. All of the structures inside of a myofibril are organized into something called a sarcomere. So a sarcomere is the contractile unit of a muscle fiber, and there's many sarcomeres in sequence along a myofibril. And then again, they're composed of these two different proteins. The thick filaments are made of myosin, and the thin filaments are made of actin. Those thick filaments that are made of myosin have something called ATPase enzymes, so they're able to break down ATP and collect energy. The myosin filaments have heads or extensions. Sometimes they're also called cross bridges, and they use those cross bridges to attach to actin filaments, and we'll talk about that in just a moment. Myosin and actin overlap somewhat in a sarcomere, and that allows them to interact with each other. And then the thin filaments are made of actin, and they are anchored to each other at that structure called the Z disc that we just looked at on the diagram picture. So if we look at one single sarcomere in sequence, remember there's many of them in a row on a myofibril. They're made of thin filaments, which are actin, thick filaments, which are myosin. The thin filaments are anchored at this structure called the Z disc, and so that's where they're attached. And then they overlap the thick filaments. So you have the thin filaments here in blue, the thick filaments are here in red. And you can see that they overlap each other, but there is a section in the middle here at the M line where they are not overlapped. That's also called the bare zone, and that is where the actin filaments can move towards each other when a muscle cell contracts. So at rest, when there is no muscle contraction occurring, in each sarcomere, there's the zone like we just talked about within the A band, and it's called the H zone or the bare zone, and it's where there is no actin filaments and only myosin filaments. The sarcoplasmic reticulum that we mentioned earlier has been specialized for muscle cells, and its specific job is to store and release calcium, because calcium is really important for muscle contraction, and it surrounds the myofibril so that there's calcium readily available for muscle contraction. So here we've zoomed in a little bit and you can see that bare zone a little bit better. So again, the thin filaments are represented here in blue, and the thick filaments are represented here in red. And remember, those are the actin and myosin filaments. And then that bare zone is where the actin is not present when a muscle is relaxed. When the muscle contracts, those two actin filaments move towards each other, and that shortens the muscle. Some characteristics that are critical to the function of muscle are here. So muscle is excitable, meaning that it is able to receive and respond to a stimulus. It's contractile, meaning that it can shorten when it receives an adequate stimulus. It's extensible, which means that it can stretch. And muscle is also elastic, meaning that it is able to recoil and resume its resting length after stretching. So muscle is very movable, but it always returns to a resting state. In order for a muscle cell to contract, it has to be stimulated by a motor neuron, which is a nerve cell that connects to the muscle cell. It's connected to a muscle in something called a motor unit, and that's when one motor neuron and all of the skeletal muscles it stimulates work together. So typically there's one neuron and it asks several muscle cells or muscle cell groups to contract together. So on this slide you can see a motor unit. In this diagram there's two. So motor unit 1 is represented by this red neuron, and you can see that it stimulates this muscle and this muscle group. And then motor unit 2 stimulates this muscle group, and this muscle group. And this allows your body to have strong or weak reactions with your muscle depending on the type of situation you're in, right? Because when you're in school, for example, and you want to pick up a piece of paper, it doesn't weigh very much. You don't need to stimulate every single muscle fiber in your arm to pick up a piece of paper, because it's such a small amount of resistance that if you stimulated all of your muscles to do that small job, it would be an overreaction. However, if you wanted to, go pick up a weight that weighed 20 pounds, you would need to stimulate more muscle fibers. So then more motor units would be involved. So those motor units allow you to change your response based on the job you're asking your muscles to do. This is what a motor unit looks like under a microscope. And so you can see this is a nerve bundle. So those are the axons of the motor unit nerve cell, and you can see where they stimulate the muscle cells. And you can see this is multiple motor units, but those are where the stimulation sites are. The neuromuscular junction is where the axon terminal of the motor nerve and muscle meet. And so we will go over the steps of how a muscle is stimulated, and then there will also be a little short movie you can watch. It's like 2.5 or 3 minutes that will be shared with you, not here. And that way you can see how those two different cell types interact with one another. In this diagram, you can see where the axon of the nerve cell interacts with the muscle. And so they interacted in an area called the synaptic cleft, and that's here in this box, and we'll look at it in more detail on the next slides. The synaptic cleft is the gap between the nerve cell and the muscle cell. The two cells don't actually make any contact, they don't touch at all, but there is a space between the two where some chemical exchange can happen. The space is fluid filled, and it's filled with something called interstitial fluid. So the action potential in the nerve reaches the axon terminal of the motor neuron first, and that's what tells the motor neuron that it needs to release some acetylcholine, and that's what's going to stimulate the muscle. To stimulate the release of acetylcholine, calcium channels in the axon terminal will open, and then calcium ions enter the axon terminal of the nerve cell. When the calcium ions enter, it causes some of the synaptic vesicles of the nerve cell to release their contents, and in this case they release acetylcholine. That's the neurotransmitter that communicates between nerve cells and muscle cells, and that leaves the nerve cell by a process called exocytosis. That neurotransmitter is a chemical released by nerve cells, and it is going to attach to the muscle cell and tell it to contract. So after acetylcholine diffuses across the space between the nerve cell and the muscle cell, it attaches to receptors on the sarcolemma of the muscle cell. When it binds to the sarcolemma becomes permeable to sodium ions. Sodium then rushes into the muscle cell, and that generates something called an action potential, and potassium leaves the muscle cell. Once this process starts, muscle contractions cannot be stopped. Now we'll go through this process with some diagrams. So the action potential reaches the axon of the nerve cell, and then when that happens, the nerve cell becomes permeable to calcium. When it becomes permeable to calcium, calcium will enter the axon through those open terminals. When calcium enters, it causes some of the synaptic vesicles to release their contents. So that's acetylcholine. You can see inside of the axon, there's all these vesicles that contain acetylcholine, and that's in preparation to be released for asking a muscle cell to contract. So when calcium enters, it causes some of those vesicles to fuse to the membrane of the axon, and it releases the acetylcholine out and into this interstitial space. Once the acetylcholine is in that interstitial space, it diffuses across the space to these acetylcholine receptors that are found on the muscle cell. The acetylcholine is going to bind to each of those receptors, which are those little green dots in this case. That's the acetylcholine. When they bind, they open this channel and it becomes permeable to sodium and potassium. The channel on the muscle cell eventually closes when an enzyme called acetylcholinesterase comes and it breaks apart the acetylcholine, which then closes the channel to the ions sodium and potassium. And then after it closes, the muscle is then ready for a new stimulus, but it won't continue to receive stimulus until more acetylcholine is released from the axon of the neuron. Once this process has begun, it doesn't stop until it has reached the whole muscle. So the diffusion of sodium across the muscle cell spreads the action potential across the entire sarcolemma so that it asks the entire muscle cell to contract when that muscle cell receives a stimulus.